se va a proceder a la presentación y defensa de la tesis titulada Hierarchy One Structural Funcional, como de Hierarchy Hierarchy One Alostérico, lanzada al doctorado del doctor, una palabra de Lucía Gregorio.
Bien, con el permiso del tribunal vas a presentar el trabajo realizado durante la tesis doctoral, cual tiene por título One Structural Function Study all of the three domain one allostéric. This presentation has been divided in 4 main parts: introduction, objectives, results, and then finally conclusions. Well, pain is one of the most common complaints with a worldwide prevalence of around 20%. So that generates a tremendous cost for global economy. So then, nowadays, why still don't we have an effective treatment for this pathology? Basically, this is due to the lack of knowledge of the molecular and cellular mechanisms involved in the physiology of this pathological condition. To answer the potential channels family, have some of the most important proteins involved in nociception. For this big family is divided in 7 subfamilies, which 3 of them has been directly involved in the detection of noxious stimuli in nociceptors, which are 3A, 3P, and 3P. Those channels respond with a very high threshold to different physical and chemical stimuli. We have a set of pungent compounds which activate these channels, but at the same time, they also respond to a wide range of temperatures going from noxious cold to noxious heat. So that's why they are also called thermosensor or thermotrips. So the fact of these channels are involved in this thermosensation and pain signaling make them as a potential target for the design of new drugs as anti-inflammatory or analgesics. But for that, it is essential to know these molecular mechanisms which are involved in their function. So, for this reason, we focus our work in study or determine molecular mechanisms involved in TRPV1 gating, since this channel was the first three-channel clone to be involved in nociception. Well, TRPV1 forms a tetramer with a four-fold symmetry and forming an aquaspore in the center of these 4 subunits. Then each subunit is formed by a 6 transmembrane segment with an extracellular core loop and 2 large intracellular N and C terminal domains. There are some important characteristics of this channel with modulated For example, phosphorylation, and for these mechanisms has been described or proposed many amino acids in these intracellular domains. Also, it exists different anchoring repeat domains in the N-terminal region, which are implicated in the interaction with other proteins. Phosphorinositides also interact directly with the C-terminal of this channel. And also has been described the glycosylation process in an amino acid placed in the extracellular core loop to be important for modulated channel. Well, as mentioned, TRPV1 is a polymodal channel responding to physical and chemical stimuli. And several structure function studies have determined the specific domains as a putative sensor for all these stimuli. For instance, TRPV1 is well known that it's activated by capsaicin, which is the hot and spicy component of the chili peppers. And its binding site has been proposed or demonstrated, we could say, in the intracellular region between TM2 and TM3. Also, protons directly gate TRPV1, by interacting with acidic amino acids placed in the extracellular wall loop. Regarding this physical stimuli, we know that temperature also directly activates TRPV1, but it has not been well or totally described the presence of its sensor. Some authors propose the extracellular wall target as to be indicated in the temperature, and others also propose this C-terminal, but there are other studies which suggest that there is not a specific temperature sensor, so that this temperature-dependent gating could be driven by different rearrangement along the whole protein. Another physical stimulus which activates TRPV1 is voltage, and positive potential drive the opening of the channel, but this voltage sensor is not well described either. So regarding other voltage-gated channels, these sensors should be somewhere in the S4 segment. So the application of all these stimuli drive the channel opening, generating over-rectifying current, and as seen in this figure, different stimuli generates currents with a different intensity. TRPV1 is a non-selective cation channel which preferably conducts calcium. Well, regarding these gating mechanisms, it is reasonable to think that amino acids close to the channel gate may be important for the gating of this channel. In fact, we found that in the C-terminal domain, there is a highly conserved sequence referred to as strip domain, which is characteristic from different strip channels of family. In the center of this region, strip domain, we also find a subdomain called tripos, which contains 6 amino acids, and it's if strip domain is highly conserved, here we could say that this subdomain is extremely conserved in different strip channels of family. So the fact of, better say, since we cannot find a high percentage of similarity between and similarity sequence between different strip channels of families, the fact that these two regions are so conserved make us to think that they should be somehow important for channel functionality. So previous studies performed in our laboratory, revealed the relevance or the importance of these regions in channel gating. First, they performed a study focused on the role of this transient receptor potential domain in TRPV1. And the strategy for this study was to generate a set of chimeric channels containing the whole sequence of strip domain, the TRPV1, sorry, and that this strip domain was substituted by the conic sequences of the rest of strip channels family. They found that all chimeras showed an impaired activation when induced by different stimuli. But the most important result, or the most outstanding result from this work, was that the chimera TRPV182 was totally insensitive to any of the stimuli applied. Even this strip domain sequence from TRPV2 was the most similar one with the sequence found in TRPV1. The main conclusion from this work was that the strip domain affects the channel coupling and protein assembly. In parallel, they performed another work, focused this time in the role of transient receptor potential, but free box in TRPV1 gating. And for this purpose, they performed nonlinear scanning mutagenesis of all these six positions, and they found that for all of them, the mechanism was allostéric. In particular, we found that two of the most affected mutations were found for the positions tryptophan and isoleucine. And from here, it is important to mention that this tryptophan position is the only amino acid which is totally conserved in all the tryptellose subfamilies. And also the position of a hydrophobic residue in this isoleucine place is also pretty highly conserved. So the main conclusion was that these two residues, isoleucine and tryptophan, were essential for channel grading. So, so far we have seen that these two regions are very important or essential for the grading of the channel. But now with this work, what we want to highlight is which specific structural requirements are needed in these important positions for channel grading, for having a correct channel grading. So, based into these works, we design our objectives, which are divided into specific ones. First, we try to identify the physicochemical features of isoleucine 696 and tryptophan 697, which are required to maintain functional TRPV1 channel properties. And the second objective would be to determine which amino acids are essential for reverting the non-functional dimera described TRPV182 to a functional state. So please, let me take a sip of water. Thank you. Well, according to these objectives, we have divided the results into different parts. First, I will talk about the characterization of tryptophane residues in TRPV1. Some common procedures to all these experiments performed at these DNAs, cDNAs are transiently transfected in Higgs cells. We perform all the experiments 48 hours after the transfection, and these batch clamp experiments are done in constant configuration. For this approach, we designed a genesis strategy where substituted each of these positions animal study to all the natural amino acids. And from this, we substituted the mutant to cysteine because of the reactivity of its sulfate group. To characterize the phenotype of these mutants, we study the activation by the polarizing voltages and by capsaicin at 100 micromolar. As it is shown in this current density graph, for the isoleucine mutants, we observed that only the hydrophobic mutants were able to maintain operational activation induced by these 240 millivolts. And also, we found that the mutation to histidine was partially activated, but could be explained by the proton form of deprotonated form of this amino acid that can simulate hydrophobic conditions. And however, when we observed the results obtained for the tryptophan mutants, we saw that none of them were activated by that high potential. Regarding the capsaicin results, we observed that for isoleucine, we observed more or less the same pattern, and only the hydrophobic amino mutations were able to respond at both negative and positive potentials. For tryptophan mutants, at variance with the responsive voltage, we observed that most of them were activated by the agonist, without affecting the physicochemical properties included by each of these mutations. So, the main conclusion here is that the substitution of any of these isoleucine or tryptophan by any amino acid impairs TRPV1 activity. So, from now, we are going to focus on some representative mutants for regarding isoleucine, we will focus on all the mutants which were partially activated. And for tryptophan mutants, we select a representative mutant of each of one of each family. Next, we analyze the impact of these mutations in channel expression. In the Western blot show the total protein fraction, the protein found in membrane by using bioaccumulation approach, and the actin that was used as a loading control. As we can see here, TRPV1 is detected as a double band corresponding with the glycosylated form, the upper band, and non-glycosylated one, the lower. So for isoleucine mutants, we observed that they were all more or less detected in the same level, and all of them reached also the membrane. However, we found some differences in the glycosylation pattern when compared with TRPV1. Regarding tryptophan mutants, we saw that all of them were also expressed and they reached the membrane, cell membrane, and there was also significant increase for the mutant trimethylene. And interestingly, none of them showed the glycosylated band corresponding with wild type. So, as the conclusion here is we can say that these mutations affect somehow TRPV1 assembly. Well, now we are going to focus on the results obtained for isoleucine mutants. We studied their functional phenotype, first by their activation induced by the polarizing voltages. For this, in a patch clamp experiment, we used a family step protocol going from -120 up to 240 millivolts. This protocol was able to evolve non-activating currents for all of the mutants studied in this case. But the current intensity was lower when compared with TRPV1, indicating that the mutations of isoleucine position to any of these amino acids alter voltage response. The analysis of the biophysical parameters from these steady-state currents revealed that the currents observed for the conductance to voltage curves observed for calculated for all mutants followed a Boltzmann distribution. Regarding the B-half value, which is the voltage at which we obtain the half conductance value, where it was significantly increased toward more positive potentials when compared for all mutants when compared with TRPV1. So, consequently, the free energy difference between closed and open state induced by this stimuli is also significantly increased. However, for the apparent gating balance, which we did not observe any significant difference when comparing all mutants with TRPV1. So, as a conclusion from these graphs, we can say that isoleucine mutants show an increase in the activation energy of the voltage-dependent agent. So, what happens with the response of to capsaicin of these mutants? To study these, the gating induced for vanilloid, we study the activation by 100 micromolar capsaicin in presence of a ramp protocol going to -80 up to 160 millivolts. What we observed for TRPV1 was that these two responses were more or less similar, getting omic behavior for 100 micromolar was applied. Also, there was an inward component and negative potentials induced by the presence of these alloys. In contrast, these inward component was lost in most of the mutants studied, except for isolers into leucine and partially to body, indicating that these mutations affect mostly the activation of these mutants and negative potentials. So, the biophysical parameters analyzed from these curves revealed that all GB curves were fitted to a Boltzmann distribution. And as observed here, we observed that still the agonist, even it generates a decrease in the B-half value, it is still higher than the one we observed for 3B1. For 100 micromolar capsaicin, we observe a linear conductance behavior derived from the omic behavior we found in the IP graph. So this increase in B-half with respect to 3B1 determines that the free energy difference between closed and open state for these mutants is significantly higher when compared with wild type. As we can see also in this graph, the mutants to valine and leucine were the most similar ones to the wild type. Well, so far we have been assuming a two-state model, but for 2B1, it has been widely described the presence of several closed and open states induced by different stimuli. An allosteric gating model has been used for explaining the gating process of ion channels containing different domains and activated by different stimuli. So that's why we try to fit this model to our experimental data. This allosteric model is based on the existence of different equilibriums, which are driven between them by an equilibrium constant, and these equilibriums at the same time are coupled to each other by allosteric factors. We have the equilibrium for the channel board between closed and open state, for the Montiel sensor between resting and activated state, and also here we add the capsaicin equilibrium for the representing and binding conformation. Also, both sensors, we contemplate the possibility that both sensors would interact between them with allosteric factor E. So, for assuming this allosteric model, we have to assume that our four sensors from the first units are identical, that they operate in unison, and that they move simultaneously. As we can see in this figure, the allosteric model fitted properly to our experimental data. In this case, we have assumed open probability equal to GG max value, since we have obtained our currents in the whole cell configuration. The most relevant data obtained from this fitting for the activation increased by voltage is shown in this graph. The apparent gating balance and energy values are pretty similar to the ones we obtained experimentally. But one of the most dramatic changes was the decrease of the factor for all the mutants included in these positions, indicating that these mutations are somehow affecting the connection between both the sensor and the channel board. What happened when we activate capsaicin? Well, we have compared the highest concentration of capsaicin applied in each case, and the most interesting results obtained from this fitting was that this capsaicin coupling factor, allosteric coupling factor, was a decrease in all mutants, so indicating an uncoupling between this equilibrium. And also for E, 3B1, we detect a different value from 1, sorry, I forgot to say that when E is equal to 1, we consider that both sensors act independently. So since we found a different value from 1, we understand that both sensors are somehow connected, but this is also decreased for these isolation mutants. Then, when we studied the functionality of tryptophan mutants, we found that the activation by capsaicin, since they did not show any response to voltage, evoked significant currents with the inward component was maintained at variance with the isoleucine mutants. Also, only for the mutant to methylene showed lower current levels, probably due to the decrease in protein expression. The biophysical parameters analyzed from these currents revealed that all of the currents induced by capsaicin for these mutants were follow a Boltzmann distribution. And interestingly, what we found was that any of the mutation included here evokes similar health values, independently of the physicochemical property of the amino acid included. There was not a significant difference between for the aberrant balance, although we found that these mutations increased almost at twofold the value of these apparent dating balance. So consequently, what we found was that the free energy difference applied by voltage was the highest component in these mutants, indicating that this voltage insensitivity is the most affected by the mutations in this position. So, as in the previous section, we tried to fit the allosteric model in our data, and apparently it fits pretty well. So, the most outstanding results we found from this fitting were that, as we observed from experimental data, the coupling between capsaicin sensor and channel 18, well, channel 4, let's say, it's more or less similar for all of the mutants studied, and it's not so far away from the values obtained on 21. The aggregating values are also very similar to the ones obtained experimentally, and the most dramatic changes were observed for the allosteric factor D. So this allosteric model also, these mutations of this position, seems that they affect this allosteric coupling between voltage sensor and channel 4. So just to sum up this part of the presentation, we can say that isoleucine mutants, isoleucine position only tolerates hydrophobic amino acids for generating functional mutants. So for that, we propose that this position should be located in a hydrophobic environment in the 3D structure of the protein. The mutations of tryptophan position evoke complete uncoupling voltage sensing, indicating that it exists the possibility that this position interact with the region S4, S5. So it can somehow interact with voltage sensor. So we have been focused so far on two specific amino acids of this region. But what happens when we study the whole context of the tree domain? In this part, we have focused on the chimera TRPV182, as I mentioned in the introduction. We contain the whole sequence of TRPV1 and the tree domain of TRPV2. I just want to remind that this chimera, this tree domain was the most similar one to TRPV1 and gave rise to totally insensitive channels. So, for this study, we designed a mutagenesis strategy where our objective was to recover the original sequence from the context of the chimera up to 3B1. So for that, we did some sequential and cumulative mutations, restoring the whole sequence. The criteria for introducing these mutations has been we first introduce what we consider the most disparate changes between both sequences. Well, so we first analyzed the impact of these channels, of these mutations, in protein expression. And what we found was that some representative chimeras are shown here, and we found that mutants 82, 84, 17, and 18 were less expressed when compared with CB1 wild type. Although they reached in a similar way, in a similar ratio, the membrane. However, it is also interesting to notice here that all of them lost their glycosylated band. So indicating that these modest changes in this three-domain region severely affect this protein expression. Then, to study the functionality of these chimeras, we first analyzed the response to capsaicin by calcium microfluorography assays. We used as a stimulus capsaicin 100 micromolar. And we can see an increase in fluorescence inducing 3B1 and some other chimeras. So then we quantified the response by using the area under the pure factor, which measures the surface between the injection cycle and the last one. And this quantification revealed that none of the mutants between 82 and 8217 were activated by this high concentration of the agonist. Only 82, 18, 19, and 20 showed activation or partial activation, getting 3B1 levels in the last one. This is just to remember which mutations are included in each of these three functional channels, these are the restored amino acids in each case, and the remaining mutations from the 82 constant. So I just want to highlight how conserved these mutations are. Well, the same behavior we found in the response to voltage for these chimeras. We applied the same family step protocol described before. So we show here some representative chimeras. We were able to create non-activating currents only for 82, 18, 19, and 20 when compared with 3B1. So, biophysical parameters obtained from these steady-state currents revealed that 8220 showed a similar, or we say equal, Boltzmann when compared with 3B1. And as is shown in these parameters calculated from the Boltzmann, we observed that 82, 18, and 20 showed significantly increase of the B-half, and consequently this the energy difference, showing no difference for the aggregating values. So the mutations included in these two chimeras, 82, 18, and 19, shows or induce an increase in the activation energy of the voltage-dependent gate. Then we studied the response of some representative chimeras to temperature. For this purpose, we used time result measurements by applying a range of temperature between 36 and 60 Celsius degrees at negative and positive potentials. As we can see for 3B1 in scent, we were able to detect currents from around 43 Celsius degrees, and it gets saturated around 53. Well, none of the three chimeras studied here, 82, 82, 4, or 14, were temperature sensitive at studied the response of 82, 18, 19, and 20, we observed the same phenotype or the same behavior as we have seen with the previous stimuli, reaching more or less wild-type phenotype for 82, 20. Also here, the presence of a positive potential induced a decrease in the temperature threshold of the activation of these chemicals. So, the thermodynamical parameters obtained from these currents reveal that 8220 showed a T-Hal value very similar or the same T-Hal value as TRPV1 has been described for TRPV1 in vivo. However, 8219 showed an increase for this T-Hal value of around 10 Celsius degrees. Here, we observe also that the presence of the application of a positive potential simultaneously with temperature did not affect T-Hal, but induced that this Boltzmann shape becomes shallower, indicating a loss of temperature dependency, of this process. So, to measure the temperature sensitivity of this mechanism, gating mechanism, we calculate this Q10 factor. In both cases, 8219 and 8220, we found that there were we can see clearly two different phases and negative potential. The Q10 value obtained in the highest, in the phase showing the highest sensitivity to temperature, were Q10, 21 more or less for 8220, which is the one we found for TRPV1, and it was significantly decreased for 8219, indicating that this chimera show an impaired temperature sensitivity of the temperature gating process. Also, by the application of positive potential, we observed that this temperature sensitivity decreased in both cases. Then, the analysis of these thermodynamical parameters by using Van 't Hoff equation from the equilibrium process between the homogeneous state revealed that 8220 show an enthalpy value of this equilibrium process of around 100 kilocalories per mole, which is the value described for TRPV1, whereas 8219 showed a lower value, around 80, indicating that this gating temperature in 8219 is less temperature dependent when it's compared with 8220. So, finally, we try to better understand the effect of these mutations are having in the gating mechanism. For that, we fit the allosteric model on our experimental data. In this case, there is one of the equilibrium represents the temperature sensor, the resting and activated state. Well, as we can see, the allosteric model properly fitted to our experimental data, and the most important or significant results from this fitting are shown in this table. In the presence of negative potential, we assume at -60, we assume that both that sensor is in its resting state. For 8219, we observed that the factor C, which determines the connection between temperature sensor and channel 4, is significantly decreased when compared with 8220. And in this case, the application of the positive potential did not affect so much this parameter at variance with the behavior, the data or the values we obtained in 8220, which was significantly decreased. This can be explained, because for 8219 we get E value of around near 1, so indicating that these two equilibria not interact with each other. Whereas for 8220, it has a value of 15, more or less, indicating that the activation of this voltage sensor could somehow affect this temperature depending rating. So finally, just to sum up our results, the main conclusion here we can say is that modest changes in the 3-domain sequence induce dramatic changes in GP1 assembly and functionality. So, I just want to mention that a couple of weeks ago, the structure of GP1 was published and by David Julius, and they used a bioelectron microscopy for obtaining this electronic mass density, and they fit this model, structural model, with this lab. So, trying to fit our data in this new structure, I know this is not similar at all with the one I showed here, but let's try to fit something. This tryptophan residue, which is the one we have been studying deeply, they propose that it's interacting by a hydrogen bond with the carbonyl group of the phenylalanine 559, which is placed in the S4-S5 segment. So we were not that far. And this indicates that changes in this residue could somehow break this interaction, affecting what is supposed to be the voltage sensor and consequently the voltage sensing, the voltage gating of this channel. The isoleucine residue would be placed in this phase of the helix. And that for us it's kind of, how to say, weird, because we have observed that this isoleucine should be a hydrophobic residue. But since they have not shown the whole C-terminal, because there are still, I think it's 80 or 90 amino acids missing for this structure, we think that there is a possibility of this C-terminal region, C-terminal missing region, which could be somewhere here. It could be somehow create this hydrophobic environment to give this isoleucine and other hydrophobic residues which are placed in this phase of the helix. So just to finish, regarding the position of the tryptophan here, which is placed here, be parallel to the membrane, and all these tremendous number of interactions with S4, S5. It goes also between S1 and S2, and also it exists, now they say that also interacts with S1 and repeat domains. It is reasonable that small changes in this region somehow can disrupt that huge number of interaction or this environment. So that's why we can say that this tryptophan is necessary that it is maintained for the converting coupling of this type. Ahora, con el permiso del tribunal, paso a leer las conclusiones. First, position isoleucine 696 and tryptophan 697 in tripbox are key residues transmitting stimulus sensing for gating through the allosteric coupling mechanism between voltage and capsaicin activating sensor and channel gate. Second, isoleucine 696 residue only can be substituted by hydrophobic amino acids for channel function. The study also revealed an effect of the amino acid size at isoleucine 696 in channel activation, since the replacement of isoleucine by a smaller or larger amino acid result in higher activation energy. Third, mutations of tryptophan 697 position to any of the natural amino acids evoke a complete uncoupling of voltage sensing and gate opening. However, this position tolerated virtually all substitutions since practically all mutants display channel activity in the presence of saturating concentration of capsaicin. 4. Isoleucine and tryptophan residues are essential in TRPV1 channel folding since discrete changes in this position evoke relevant changes in the protein expression pattern and glycosylation process. 5th, straight domain integrity determines gating properties of TRPV1 since modest changes in this sequence severely affect coupling properties of channel gating between the voltage and temperature sensor and with the channel core. And 6th, straight domain plays an important role in channel expression and folding since mutation within this sequence dramatically alter protein expression pattern and glycosylation process.